next Thursday we have an exam, right? We're only going to get to practice problems that we did yesterday in discussion, and then next week is a whole different topic. The most important problem, I think, is thinking about selection, because that is how we can see evolution acting in contemporary population. So if you want more practice on that, we'll do a very small example in class today. But if you want more practice on it, where should you look? There's always so many practice problems in your book. So every chapter has work problems. So here's a work problem. It's the same system that we saw yesterday with AH alleles and Drosophila, but the numbers are very different. The highest fitness here is the FF homozygous. So it has a solution strategy. So I would suggest if you're wanting more practice on this type of problem, do it by yourself, then do the solution strategy. There's also always the assessment at the end of every chapter. So I haven't looked at this yet. But if you go down to applied questions, you can always get more practice, right? You can get practice thinking about allele frequencies. You can do some selection. Right? All of these are, you can get practice doing a chi-square. But all the same problems we're doing in discussion, we just don't have the time to assign so many things. So if you know you need more help with chi-square or selection problems, try these. Talk to people, come to office hours with me or Dr. Groovy, so that you're confident going into the exam about these types of questions. Any questions on this? We'll do an example today. Really quickly though, right? We don't have that hour to do one example at the beginning, but okay, so today we're gonna kind of finish genetics of natural populations in terms of population genetics, and then Tuesday we're gonna scale up to thinking about how evolutionary genetics help us think about relationships, not between populations, but between lineages and species. So you have a draw to learn. There's been really interesting studies, like selection isn't something that just happened in the past, it's something that's ongoing, and you can measure it in real time. There's been tons of really interesting studies using Paramiscus, so they're a genus of mice, where you can see strong selection based on local environments. So there's dark-colored mice, light-colored mice. It correlates with the sand color in Florida. People have put like fake mice out, and if you mismatch them, they get attacked by hawks or snakes more easily. Studies like this have also been done in Nebraska, in the sandhills. So I know some of you are from Nebraska, some may not be, but western Nebraska has some really cool natural areas called sandhills that replicate this. You have dark and light soil. So there's a draw to learn. You're going to think about mice undergoing selection to help you think about those selection problems, because those are so key and important to what we do. So where we left off last class was thinking about Hardy-Weinberg equilibrium is our null model. When there is no evolution, allele frequencies and genotypic frequencies do not change. If we violate that model, something's interesting happening. It's evolution. So if we define evolution as the change in allele frequency, there are several forces that can change allele frequency. So these are like the conditions of Hardy-Weinberg, and we violate them. So instead of no mutation, we're now getting mutation. We're now getting something other than random mating. We're seeing natural selection. We're seeing small populations that are more vulnerable to stochastic sampling, and then we're seeing gene flow. So this is migration. So we're not going to go through these in order. We will start with mutation, and then we'll kind of skip around, leaving selection at the end. Do people care about Hardy-Weinberg equilibrium? Do people actually do this? Yes, people do this all the time. Of course, in human genetics, we are not doing an official, formal highlighted scientist, because you should be focusing on the writing assignment. But some of my favorite work that I've seen from like really exciting young scientists are population geneticists. So I just had a couple here. So most of these are people that I've met or know. So I kind of cherry pick them, but they're doing really, really cool work. But it won't advance my slides. So Nancy Chan, she's a professor at the University of Rochester. She works on this organism called scrubgeons. They are endemic to Florida. She combines population genetics with pedigrees. They go down to Florida every year and match each individual bird and all their offspring. And what can you do with that? You can understand how allele frequency changes over time. Where else do we use pedigrees a lot? In human population. Even though she's studying a bird, she has making really important insights into human health. And so that's how she gets funded to do this really cool work. Jaslyn Beauty is at the University of Southern California. She works on dogs and wolves genetics and inbreeding. Inbreeding is also very important for disease traits in humans. So they're using these cool systems, but making fundamental discoveries about biology. And Ellen Leffler, she's at the University of Utah. She does 3 organisms. So picture there is Plasmodium, right, that contributes to malaria. So she does population genetics of Plasmodia, Anopheles mosquitoes that carry Plasmodia, and then humans. So how are all 3 kind of co-evolving using population genetics? So it's not just kind of this old thing that we think about. It's kind of ongoing, has super important implications for these systems, but human health and disease as well. So I was supposed to have a slide on mutation, but it looks like it disappeared. So we did mutation last class really, really quickly. So I'm not going to do it again today. We're not going to see mutation again. I wanted to start with mutation because we talked about mutations a lot in genetics, and it's one that we can hopefully build an intuition, right? We have a big A. Let's say that there are no little a's in the population. The only way to get little a would be through mutation. And we can describe that system through mutation. So we're not going to see that again, so I guess my slide missing isn't a big deal here. So now we're going to move to genetic drift. So this is our kind of second violation of Hardy-Weinberg equilibrium. If we have a really large population, there still is sampling. There's still a stochasticity, but you don't see it as often as you do in a small population. So when you see changes in allele frequency that are just due to sampling processes, this is what we call genetic drift. So some people want to say chance. I'm fine with that, but I like sampling better. Chance, I think of probabilities. Yeah, probabilities are different in small populations. I still like to think about it as sampling. Why is it more common in small populations? Because, right, that's like, there's a statistical property that has to do with the scaling of variance and sample size. But you can think of it just like flipping a coin, right? We know that the probability of flipping heads or tails should be 0.5. If you flip 3 coins, the probability of seeing all heads is actually quite high, right? Just because of the small sample size. The probability of seeing all heads when we flip 1,000 coins is very low. That's what genetic drift is doing. It's kind of sampling, and you see kind of probability shift, not due to anything real about the changes in the probabilities or selections, but just due to the small sample. So there's kind of, how do you go from a large population to a small population? There's many ways that can happen. Some of them are ecological, lack of resources, but the one that your book talks a lot about is called like a founder effect and bottlenecks. So founder effect, you can think of it as like something from a large population on a mainland going to an island, right? Oftentimes island populations are colonized by 1 or 2 individuals. So whatever alleles that they have, they're bringing to this new population just by chance. It's not because those alleles are better on an island, it's just chance. You sampled a few individuals that made it themselves. Bottlenecks happen a lot too as organisms or lineages go through range expansion and range contraction. So there's a really famous bottleneck. Can anyone think of a bottle? Maybe you don't know it like this, in this term, but there's a really important bottleneck that kind of affects most of us in this room. That you're taking a large population, you're making it really, really small, and then you're blowing it up again. I think I mentioned this before. Human population, right? Out of Africa migration, When human lineages came out of Africa, they went through a severe bottleneck. That's why there's so much more genetic diversity in Africa compared to out of Africa, just through sampling. So bottlenecks have really important implications for most of the human genetics in the world. So for genetic drift, we often don't calculate changes in allele frequency over time because it is so stochastic. What we care about more here is variance. So we've seen variances like sigma, so this is variance. So the variance in the allele frequency is equal to the frequency of kind of heterozygotes over the total sample size. You don't have to remember this. The point here is that for genetic drift, we care more about variance. That's the metric that's going to tell us something because it scales with population size. And you can see that from this equation, right? If n is our population size, as n gets larger, variance has to get smaller, right? So we multiply by 2 because we care about the number of chromosomes, not the number of alleles. And the max allelic diversity is when 2 alleles are at 0.5, right? That's when you have maximum diversity. So that's how we measure genetic drift. So this is just an illustration from your book about a bottleneck, like literally using a bottleneck, right? If you have a lot of genetic diversity in this bottle, but only blue and yellow marbles come out, then as the population increases, it's going to increase allele frequency, blue and yellow, for no other reason than sampling. So what does that do to populations over time? It reduces their genetic variation, right? If you start, and people, mathematicians had shown this a long time ago. They had derived that variance equation. But experimentally, it's actually hard to show that. So this is an experiment explained in your book. So they start with a visible marker in flies. So they said P is 0.5 for a visible marker known as brown-white. And then they let these generations go at a small size over and over and over. So at the beginning, right, so 1 is the beginning of the experiment. Most populations stayed at 0.5. Then just through sampling, no selection, this is in the lab, they drift. So that's what it means by drift. Instead of staying constant, you drift. So each part of the histogram here is an individual population, or it's the summation of how many populations show that allele frequency. But if you drew an individual population, right, they start here, and they kind of drift around, then they fix. So this is really important. Fixation is an equilibrium, right? Genetic drift reaches over time an equilibrium. Equilibrium, we said, is when the allele frequency is not changing. Artie-Weinberg is just one special case of an equilibrium. Genetic drift often reaches equilibrium. So over time, right, most of the populations here fix. One allele, either P or Q, either big A or little a, just due to sampling a small population. So if you look at all of the populations, if you look across all of them, the allele frequency is the same. The allele frequency is still 0.5. Just in an individual population, they fix. So important in the sense of drift is changing things stochastically. It's not important that you don't have to remember what's happening. Any questions on drift? So we've done mutation, we've done drift. Next, we're going to do non-random mating. Non-random mating happens many different ways. There's a lot of definitions of this, but really they all still follow kind of one type of population genetic model. So we'll define some of them and we'll see an example, specifically big breed. So we have positive assortative mating. Negative assortative mating. And then inbreeding. So does anyone want to take a guess at what some of these mean? You can either shout it out or put it in the chat.
Positive, it refers to the tendency for like individuals to mate.
Yeah, exactly, yeah. So we can think of that at a phenotypic level, right? There's a trait that you have that someone else has, right? But we want to think about it at a genetic level too, right? We assume that similar phenotypes have similar genotypes, we can define it like that. So similar genotypes are more likely to mate. So this is somewhat easy to show in certain animal species that have really conspicuous, like binary characters. But it's been shown in humans too. Whether you believe the studies or not, I don't know. But like, there's a lot of things about like facial symmetry and how you recognize your own face that can choose mate choice in humans. I don't know if I believe all of it, but there is some evidence that this happens in humans. It definitely happens in a lot of animals. So then negative would be the opposite, right? So this would be dissimilar genotypes would be more likely to mate. So here it would be like we're doing big A by big A, right? Genotypes that are the same preferentially mate. Here it would be dissimilar. So you get matings like that. And it's going to be way more complex than kind of single locus dynamics, but hopefully that will illustrate it in terms of genotypes. So then what's inbreeding? I heard something, but I don't know if it was sarcastic or not. And I couldn't really hear all of it. Yeah, so inbreeding is when you mate with an individual who's more genetically related to you than the rest of the population. So that's mating between individuals that are genetically related. You might see inbreeding more frequently in a positive assortative case, right? If a negative assortative case, we have big A, little a, they often have different alleles. It's unlikely they're related. Big A, if they share those big A alleles through identical by descent, which means they share those genotypes because they share grandparents or something, that's a case of inbreeding. So what's really interesting about inbreeding is that we include it as a kind of force that violates Hardy-Weinberg equilibrium. But it doesn't actually change allele frequency. It only changes genotype frequency. And this is kind of hard to see unless you kind of like make a pretend population and kind of track it over time. That's the only really way to see it. We'll see it when we do an inbreeding problem in a little bit. But you can think about this, right? Think about this situation. In Hardy-Weinberg equilibrium, what would our genotype frequencies be?
0.9 and 0.49.
0.09, 0.42.
So now let's assume that, so this would be Hardy-Weinberg equilibrium randomly. Now let's assume there's a case of positive assortative mating. Let's assume that big A only mates with big A, and little a only mates with little a. Our frequencies would look like this now. So this was Hardy-Weinberg. This is positive. Allele frequencies are unchanged, right? The allele frequency here, you can't just take the square root. We're no longer in Hardy-Weinberg equilibrium. But the allele frequency of P is still 0.3. The allele frequency of Q is still 0.7. So this is like an extreme case, but hopefully this really gets at the point that genotype frequencies change, but not allele frequencies. So this violates Hardy-Weinberg equilibrium, but, right, we can no longer use the binomial expansion to classify this population, but it actually doesn't lead to evolution as I've defined it in and of itself. Any questions on that? That's kind of, it's kind of abstract, but hopefully this example illustrates this. So if you have to remember one thing for non-random mating, it's that. It's the only force that's going to not change allele frequency. Okay, let's do this problem with inbreeding. Inbreeding, because it's not changing allele frequency, it's only changing genotype frequency. What did we see happen in this, right? You actually saw fixation of homozygotes. You only see big A, or little a. That's a problem when you have recessive diseases. Inbreeding we saw in pedigrees was really the only way to get autosomal recessive diseases seen frequently. And so that's because you're fixing homozygous genotypes. So it's going to lead to the fixation of homozygous genotypes. And if they happen to be recessive disease alleles, you'll see an increased frequency in disease. So there's an inbreeding, you can classify for like an individual or for a population the level of inbreeding. We're not going to do that calculation, but it's important just to think about the coefficient and what it means, because that's how we'll apply it in some of these problems. So we can measure that with the inbreeding coefficient. F, and that ranges from 0 to 1. So 0 would be random mating. And 1 would actually be like, 1 is a level of inbreeding really hard to reach in populations that have separate sexes. It's almost like self-fertilization. So plants and some insects and nematodes can get up to an F index of 1, but like things that have separate sexes, an inbreeding level of 1 would be really difficult. So you don't have to calculate that, but you should think about it. So low level, random mating, high level, really extreme case of inbreeding. So what are our equations that we want to pay attention to? We don't want to track individual alleles. I said those will not change. We want to track genotype frequencies. So we're going to have, right, in the next generation. So in the next generation, we can define these. So if we think about the big A homozygote, we take its frequency, we take heterozygotes, but with an inbreeding. We assume that there's kind of positive sort of mating. Heterozygotes are going to mate more like with heterozygotes, but we weight this by that F, that inbreeding coefficient. Right, it's symmetric too. Inbreeding fixes, right? It's going to increase the frequency of homozygotes. It's going to decrease the frequency of heterozygotes. So that's why you see the minus FEQ in the heterozygote equation. So that's how we've defined inbreeding, and it shows up in these equations. So if you took the equations we were using Hardy-Weinberg equilibrium and put in F, this is what you get. If you took like a binomial system that expanded it, this is what happens. So I think this should be our first pop tax question. So you're studying an isolated population with an inbreeding coefficient of 0.35. There's a recessive allele that encodes a disease in the frequency of 0.01. What is the expected frequency in the present population? So assuming Hardy-Weinberg equilibrium. So this frequency, that's the phenotypic, but that's the disease frequency. That's not the, I'm asking you for the frequency of the allele, and I'm giving you the frequency of this trait. Oh, I didn't, no, I'm asking, that is the allele frequency, sorry. I changed this question yesterday, and I didn't realize it didn't make it into this slide. So the frequency of R, so what is the frequency of the genotype, the homozygous? Apologies for that. I'm seeing a lot of different answers, but some of that might be because I messed you up, so I'll give it a second to re-equilibrate. If we assume Hardy-Weinberg equilibrium, our binomial expansion holds, right? So we can just say R squared. So that's like 1 in 10,000. You know, it's not a super rare disease. It's not super common either. So what happens when we add inbreeding? So now use your F coefficient. How many RR homozygotes are you going to expect? So I'll remind you what that equation looks like. Right, the frequency in the next generation of a homozygote was the frequency in the current generation, inbreeding coefficient, those guys. So the number that we already have right? So we're just going to plug it in, but we should be able to make a prediction. It should increase. Like even before plugging something in, we should think about how this allele and the genotype frequency is going to increase, right? Because we're adding F and the heterozygotes. There's going to be more heterozygotes than homozygotes because the allele frequency of little r is so small. So by default, this has to increase. And then we can show that with the equation. That's what I got. Does that look right? So it's still a small frequency, but it's actually 5 times higher than the previous.
6, I can get 6.
So that's really, really important. Inbreeding doesn't always have to be a bad thing. Plant breeders, animal breeders use it all the time. If you inbreed an animal and it doesn't suffer from what's called inbreeding depression, that would be carrying these recessive alleles that now cause something bad. If you can make an inbred plant that's healthy, you eliminated all the bad stuff. And now you have like the starting block and you can do whatever you want. The problem is if you have only one genotype, you can't have all, right? If you're all little a, you can't can't evolve, right? You can't change allele frequency to big A. So oftentimes inbred populations in the context of breeding, you kind of at the end of selection. If there's an environmental change, you're screwed. Same with conservation, right? An inbred population of like panthers in Florida doesn't mean it's bad. It just means they might be more vulnerable to climate change. Any questions on inbreeding? Take home. Non-random mating does not change allele frequency, only genotypic frequency. Okay, so migration. So this was a homework problem. We also did migration in class yesterday, so hopefully this will just provide more context for what we did. Migration is going to alter allele frequencies by making two populations look more similar. So again, we can make a prediction. Before we do any math, we should be able to predict what's going to happen. If the allele frequency of blue is high in population 1 and low in population 2, and the new population, it should be in between. Not like exactly in between, but it should be in between, dependent on how many migrants are being exchanged between these populations. So when you hear migration, you might think of individuals moving. That's true, but we care about alleles moving, right? So migration is individuals moving. But for population genetics, we care more about alleles moving. Why is that differentiation? Why is that important? Does anyone think about why that's important? Any ideas? Maybe I didn't explain well. We care about alleles that actually make it to the next generation. So does that imply something?
Introspection.
Say that you're a person living in London. You migrate to the island of Jersey. Are you just going to fit in naturally? Right? Jersey is a small island in between France and England, in the English Channel. They have a whole different culture, right? Even though they're sort of English, they have a whole different culture. Migrants don't always find mates, and they don't always pass on alleles to the next generation. So that's why it's really important. Like, at some level, we have to estimate migration through individuals, but for population genetics, it matters that they pass on those alleles. They successfully find a mate and reproduce. If they don't find a mate and reproduce, who cares that they migrated? They didn't affect the population in any meaningful way, in terms of population genetics. So this is the equation we worked with yesterday. This is like the most general equation. So we're seeing another prime, so Q prime, the frequency of this allele in the next generation. We have to assign populations. I think of, you can assign these populations however you want. Yesterday we saw like inland and coastal. You just have to be consistent internally. You have to say that the population you're considering is like receiving the migrants. Otherwise, you'll get the direction wrong. So here, direction is really important. So you're receiving migrants from this other population at some frequency, at the frequency that the migrants are making up this new population. Okay. So M is like the migrant number. Divided by the population, but it's the new population. And then what else? You're adding kind of the rest of the population, the population that was already there that now received the migrant. So they make up the rest. So it's kind of like migrants, non-migrants, making a new population. So we used this equation yesterday. Some of you were using a different algebraic rearrangement that may be found on Google. But I prefer this because it makes you think explicitly about the direction tied to that migration coefficient. So we have 500 individuals from a population with this allele frequency. They migrate and join an existing population. What is the new FA in this population? So how am I going to set this up? So this one's not in top half, the next one's in top half. So what do I need to know? What is the problem giving me, and what is it not giving me? When I look at that equation, I'm missing something. What am I missing?
Migration value?
Yeah, I'm missing M, so we can calculate that. So M was migrants divided by new population total. The migrants should be present on the numerator, that's who they are, but they're also on the denominator. They're contributing to the size of this new population. So 500 over 3,000, I don't remember what that is off the top of my head. Now we can use the equation. Now we can plug in this.
0.166.
So what is our starting, where did the migrants come from? They came from an allele frequency like this, right? And the remaining population has this allele frequency. It's in between. It's not exactly in between. It would be exactly in between if the number of migrants equals the number of source population, but it's kind of weighted by this migration coefficient, right? We had a population at 0.2, we had a population at 0.6. We're kind of making them look more similar. We're homogenizing them. So now the population's at 0.53. You added new individuals who had a lower frequency of the low A allele. So the overall frequency of that allele in the whole population is now also low. Okay, so I think this is the top half question. I probably wrote it on top of it. So now if we're back in Hardy-Weinberg, what's our frequency of heterozygotes? So it's really important to be able to go back and forth between alleles, between genotype frequencies, and knowing when you're in and when you're not in Hardy-Weinberg, when that binomial expansion applies and when it does not apply. So what equation are we going to use here? What are we going to use to calculate the frequency of the heterozygote?
2 pi q.
Mm-hmm. And we're back in Hardy-Weinberg. Our binomial expansion tells us that the expected frequency of heterozygotes is 2 times the 2 allele frequency. So if you did the math on this, you can actually show that the allele frequencies do not change between here and here. What changes is genotype frequency, right? Once you're in Hardy-Weinberg equilibrium, you're no longer changing allele frequency. You're kind of restoring the relationship between allele frequency and genotype frequency. So you could show that if you wanted to. Any questions on migration? Sometimes the impacts of on migration last a really long time. It's not something that just happens and it's over. migration, if alleles are successfully introduced into the genome, can be passed down thousands and thousands and thousands of generations. So has anyone done a DNA test? No? No one? You guys are smart. You don't want the man having your DNA. Smart. When human lineages came out of Africa, there were tons of other hominin lineages around, right? There were, in Europe, there were Neanderthal, Asia, there were Denisovans, right? We weren't the only hominids existing. And there was gene flow. Like, it persists. I have a little more than average number of SNPs that share common ancestry with Neanderthal than most 23andMe customers. Most 23andMe customers happen to be kind of white Europeans, so, take that with a grain of salt. But if you have a lot of African ancestry, you'll probably have very little Neanderthal DNA. If you're Asian or East Asian, you also have very little Neanderthal. It's mostly kind of people with ancestry of European.
With as much migration that has occurred for humans over the centuries, how are these companies able to pinpoint these locations? Because they get very specific.
Yeah, I mean, one, so if you look on like 23andMe, it has a confidence interval. They get very specific if you say you're 50% confident. If you crank that up to 90% confident, all of that goes away. But there is still a lot of historical population structure, right? Like indigenous people to the Americas, you can pinpoint that really, really accurately. People in African heritage, you can't pinpoint maybe where, but you can pinpoint that really well. Like, yeah, can you pinpoint you came from this province of Germany? No. Like, they just do that to sell you stuff. I forget what I was going to say about that. My point, though, is that it exists. And we'll talk about it kind of next class with phylogenetics, but it has really important implications for human health. A lot of kind of good traits that humans might have needed are now actually detrimental. So there's a lot of disease traits that are associated with Neanderthal. How do you know they come from Neanderthal? We'll talk about that next week, because we'll talk about phylogenetics. You can trace identity of genes, SNPs, whatever you want, by looking at relationships of DNA sequences. So I don't have a lot of Neanderthal. My PhD advisor had like more Neanderthal than like 90% of the population. She's Australian, British, so like, right, she's all white. She's whiter than me. So. Okay, the last course that we're going to talk about is natural selection. So, right, things don't just evolve like that. It takes time, but we can see selection operating in real time. And what did you see last class or discussion? Natural selection is all about fitness. So fitness is just kind of a catch-all term that we can measure relative, from one genotype to another that catches survival and reproduction. Selection can operate on individual traits, right? It can operate on like drought tolerance, but only if drought tolerance is increasing fitness. So there's kind of like this indirect effect. Selection operates on traits, where we see traits change, but only if they impact fitness. Right? That's why you cannot select against Alzheimer's. Most people who have Alzheimer's have already reproduced. So that's why some of those diseases that happen kind of in older populations are so persistent, because they've already reproduced. Right? Evolution can only act on fitness. It cannot act on things that are post-fitness. So traits and increase fitness, and alleles can increase or decrease in frequency. Sometimes we know about the genetic connections between an allele and a trait and fitness. Other times we don't. Right? There's some traits that people, animals, plants have, but how does it actually translate to fitness? That's a really big open question in kind of evolutionary biology. Another really important thing about fitness is environmental dependent. I showed that to you with the mice, sort of, right? I told you that mice co-occur on soils where their color matches. That's because fitness is environmental dependent. If you're a dark colored mouse on dark colored soil, you have high fitness. If you put that mouse on low colored soil, you have low fitness. So fitness is kind of like heritability. It's only important in the population you're looking at in one point in time, right? What if there's a beach that now gets washed away and it's all light soil? Okay, so time and space are really important in determining fitness. There's no like one genotype that is most fit. Very rarely. There's a lot of different types of selection. I'm not going to go into this in too much detail. Your book talks about this a lot. But I wanted to talk about 3 types just because you mentioned how kind of variance changes and allele frequency changes, and you can think of it like this too, right? So I'll show you the equations in a couple slides, but say we have a population that looks like this. So this would be a phenotypic distribution. Selection can act on this phenotypic distribution in many different ways. We can get directional selection. Disruptive selection. And stabilizing selection. So this is phenotypes, but it's going to change allele frequency. So does anyone, for any of these, have an idea of what the phenotypic distribution is going to look like? Let's just give this a number so I can do that right. I'm going to draw means and variances. Does anyone have an idea of what should we define what these are? Directional selection. You're selecting in one direction. You're selecting for an increase of this trait, which means an increase of the allele that underlies that trait. Disruptive selection. You're selecting in 2 directions. For some reason, selection is favoring maximums and minimums of the trait value. Stabilizing selection, you're selecting for the mean, strongly for the mean. Any idea what this will look like?
Directional means should be towards one side, additionally of the palm hill.
Yeah, we're going to shift. Right, we moved this way. We moved the mean. We also decreased the variance.
Disruptive is binomial.
Yeah, 2 bimodal, 2 modes. So mode is like piece, right? So we didn't shift the mean, actually, but we increased the variance a lot. We have 2 means now, sort of. The overall mean didn't shift. What about stabilizing selection?
The mean with less variance.
Yep. Do you have to remember that? No. The important part for me is that you keep thinking that, yeah, we're so focused on these alleles, but there's traits, right? That's what genetics is so important about. We can connect fitness, traits to alleles. This is what the phenotypes would be looking like under the different selective scenarios that we're going to discuss. So what's really important about fitness? It's that it's relative. It depends on the environment. It depends on space and time, but it depends on the fitness of other individuals. It doesn't matter if my fitness at an arbitrary value is 1 million, right? That sounds like a big number. That doesn't matter. What matters is the fitness of my genotype compared to other genotypes. I might be at 1 million, but another genotype might be at 2 million, right? Numbers are arbitrary in terms of fitness. What we need is a relative scale. And you saw this yesterday as well, right? If we have genotypes like this, with fitness values, this, we don't care, or sorry, a trait value. We don't care about the absolute value, we care about the relative value. So what do? We take the trait values and divide by the individual with the highest trait, if we assume that's the highest fitness. So this would be 10 divided by 10. These are important because these are how we're going to scale allele frequency changes in the subsequent generation. We've also seen those equations, right? We're eventually going for something like this. We need to think about the change based on the current frequency and fitness. So some people often misquote, right, like survival of the fittest, to say like people who are stronger, faster, blah, 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 blah. That's a horrible misquote. Survival of the fittest does not mean those traits might be related to fitness. The survival of the fittest is who's coming up with more babies, right? So when all those other hunter-gatherers were off, the male with the most fitness was the one sneaking around the tent, right? So these are what the different fitness, these are how you map kind of fitness onto selection that we define, right? So I drew these trait distributions. Directional selection means that there's one genotype, this is kind of the flip side, one genotype is unfit, W22 is really unfit, but it's like saying there's one allele that's really fit, so there's one trait. So you can have directional selection in both cases, right? The example we saw yesterday in class with the softball or discussion, sorry, with the ADH alleles was an example of overdominance. You don't have to remember anything about these, really, except they might help you think about how allele frequencies will change, right? If you know that the big A or A1 is the most fit, you should expect an increase in that allele frequency, right? If the heterozygote is the most fit, you should also expect an increase in allele frequency, but you have to take into account which is like the next fitness in this hierarchy. So it looks like a dominance hierarchy, right? I think there was actually a question in one of the discussions yesterday. Like I'm getting these traits, but I don't know about dominance. I think someone told me that. You do know about dominance. You don't know about dominance for flower color or bristle number, but you know about dominance for fitness. So if these look like dominance hierarchies to you, it's because they are. The dominance now is in terms of fitness. So I would give you this on I think I have this exact thing on your exam sheet, or exam 3. You don't have to memorize it, but you have to think about what's happening, right? So the first row is current frequency before selection. The middle row is fitness. We've now made it relative. The third row is kind of the proportion that you're contributing to the next generation. You'll see that these kind of numerators are recycled, right? So when doing these problems, I like to set them up in a table like this. It helps me kind of keep track of what's happening to each genotype through the selection process. So it's these last two rows where selection is being applied. So the third row is, okay, here's my frequency. This is the number of offspring I'm making. The third row takes into account that my fitness is only as good as the average fitness of all the other genotypes. And then it gives you, well, what is average fitness, or W bar? It's kind of the sum of all these numerators. Right? So it's the sum of the third row. And then you can find the frequency here. So it's in really small letters at the bottom. But I can find the frequency of the P allele in the next generation by transitioning from genotype frequencies to allele frequencies. Right, these are genotype frequencies. And this is the allele frequency we care about. You don't have to call them P and Q. You can call them F and S if that's the allele that you're working with. It's super, super important to be internally consistent when you're doing the math. So we're going to do an example problem now. We're going to walk through one of these. So we're going to say that's a 1, that's not a prime. It just looks weird in that font. These are our initial allele frequencies. And these are our fitnesses. So what do we need to do? Sometimes you won't have fitness, sometimes you'll need to calculate fitness. But what are kind of the next steps of what you need to do?
Well, A1 is squared, so A2.
Right, you have to think about before and after. That's what this table is telling you. Row 1 is before, row 4 is after. Before selection, after selection. So in the first population, in the first time, we're assuming Hardy-Weinberg equilibrium, right? My allele frequency, my genotypic frequency, the frequency of AA is what? We already did
109.
this, right, in our previous slide. I can use the binomial expansion before selection, because I'm in Hardy-Weinberg equilibrium. After selection, I'm also, you can assume that, but you can't assume that that transition is holding up. That's when selection. Right, so the before is a genotypic frequency based on Hardy-Weinberg equilibrium. The after is a genotypic frequency based on fitness and selection. So let's do this really quick. I don't think this is a tough question. No, I think there's another tough question. Okay, so let's do this. So what is the frequency going to look like, right? Let's start plugging in these numbers. Right, I need to know the frequency in my current. I'm only going to do the heterozygote. Well, I have to do them all, I guess. What was this, 4-2?
Yep.
Did I mess that up? No, good,
No,
okay.
that's good.
And what else do I need? I need average fitness, right? So I've done row 1, check. Row 2 is already given to me. I need row 3. I need to think about how these alleles are going to be passed down due to their own fitness, and I can use those values to then get W bar, my average fitness. So W bar is going to be 1 times 0.09 plus 0.5 and 0.42 plus 0.2 times 0.1. So these correspond to kind of the 3 different columns. The fitness of the A1 heterozygote and its frequency kind of marching down. Right? So that gives me W bar and gives me these guys. And then I just need to calculate the last one, the actual genotype frequencies when I take into account fitness across the whole population. So this would be, and we're recycling these, right? So this is the next generation now. In terms of genotype frequency, right? That's where we have 2 of them. So this is 1 times 0.09. What is it? I think the denominator turns out to be like this. So right away, you can see a huge impact of fitness. Big A was at 0.09, now it's at 0.226. So that's not an allele frequency. The problem's asking for an allele frequency. But right away, you see what we predicted. Big A has the highest fitness. It should increase in allele frequency. It does by a huge amount, right? You technically, if I'm asking about big A, you don't have to do little a. I would recommend always doing all 3 because you can check, right? They should all add up to 1. They all added up to 1 here. After selection, they should all add up to 1. Frequencies always have to add to 1. So I'm not going to do the last one, but you should, as a double check kind of on yourself. It's kind of like when we do a chi-square, we want to make sure our expected add up to our observed. You always want to make sure your genotypic frequencies are adding to 1. Your allele frequencies are adding to 1. If they don't add up to 1, something bad happens. So then the final allele frequency is 0.226 plus 1/2 because we're dealing with the heterozygotes with private traits. So the allele frequency jumps from 0.3 to 0.49. That's a pretty big jump in a single generation. So I think the top half question is supposed to be for a different slide, but I want to do it for this slide. So that's one generation. So we're going to ignore the top half in this coming slide. We're going to answer this question now. What's the equilibrium frequency of this allele? This might not work if it's expecting a number. You can, if you're thinking of a word, translate that to a number. You can't do more math. You just have to think about it. More math will not get you to the answer. So this is assuming that selection stays the same. So we did one generation of selection, but it's going to happen over and over and over again with these same fitness values. What's the equilibrium going to be? So if you're quoting 0.5, we're not in Hardy-Weinberg equilibrium. It's not going to stay the same. We're going to apply selection over and over and over. While you're answering that, I can answer questions. Any questions on this equation? Where the numbers came from, why we did it this way?
Were you just given the frequency for the like, was that given?
No, I was given the frequency of the A1 allele, which I'm calling big A, and then I had to assume before selection Hardy-Weinberg, so I squared that number. So 0.3 squared is 0.1.
Okay, gotcha, thank you.
So I used this information to get these unit frequencies using E squared, E2. I was given fitness. Oftentimes you're not given fitness. You have to calculate fitness, remembering it's relative from a trait value. Okay, so I think only 20 out of 60 have answered this top hat question. So I'm going to leave it open a little bit. Any more questions on this? Practice this. It's really important. What do I mean by it's really important?
It's on the
It's on the
exam.
exam. You will do this on the exam. So if you kind of can do it mechanically, great. But I really want you to think about it. Why is this happening? Okay, answers are still slowly coming in, but we need to move on. I only have a couple more slides. Does anyone want to answer what's the equilibrium frequency of A? Of A1? Yeah. Or was that your answer too? Yeah. It's going to fix. It's going to go to a frequency of 1. It's going to keep in, so what's equilibrium? Equilibrium occurs when the allele frequency is not changing. It's going to continue to change. It's going to keep climbing till it fixes, right? It's going to hit that boundary of 1. So the equilibrium frequency of A1, if we just do selection over and over and over, is to keep climbing higher and higher and higher until it's fixed. So the answer to this problem, the best answer here is 1. It's going to be present in all individuals. There will be no A2. There will be no little a. This allele will fix.
I
Is
didn't
that
put it
really
in the
likely?
merit form.
No, not really. But if you just kind of follow the math, that's what will happen. Any questions on that? I wish I could. I'll draw it here and then erase it. Starting here at 0.3, we saw just after 1, this is time and generation. You guys actually saw this in Draw to Learn week 1, right? You're supposed to draw something like this. Meiotic driver over time. In 1 generation, we saw a huge leap. It's going to go then asymptotically plus 1, and then 6. So that's fixation. it's gonna be really steep, and it's gonna slow down, because most of the other individuals will have the same fitness, and then it's gonna switch. Okay. So this last couple of slides, the goal is to combine things. There's no population that's only experiencing drift, or only experiencing mutation, or only experiencing selection. All populations are experiencing all of these forces simultaneously. So we're just going to take one example, mutation and natural selection. How are those contributing, right? If selection is increasing allele frequency, or trying to decrease it, the mutation acting in the opposite direction, what's that going to look like for allele frequency change? So we're going to do this scenario. So selection. Another word you could use here is be deleterious. So your book talks a lot about neutral theory, nearly neutral theory. We haven't talked about that. The words that go with that theory are deleterious or neutral. So you can call it either way, but what does that mean? It means it's the least fit allele, right? The negative effect of this is fitness. The detrimental means the fitness of this allele as well. And then we're going to have mutation from kind of wild type to this deleterious allele. So does anyone have a thought about what this might look like under equilibrium? So how do we write equilibrium? So we're going to have an equation, but what if we had to write this in words? Picture kind of like the graph I showed you. Like there's going to be a line over time. We're going to reach an equilibrium. Something's going to be pushing this line in different directions. So what's going to try to be pushing this line down? It's stable, it's staying the same, but there are things happening. So what's going to, given the definitions I just showed you, what's going to make this line go down?
Low fitness.
Yeah, selection, yeah. Selection is going to be trying to drive this allele down, but there's going to be constant recurrent mutation driving the frequency back up. So it looks flat. It looks like it's at equilibrium, a state of no change. So if we had to now think about what are our two parameters. Mutation we measure with mu. Selection we measure with fitness. We also talked really briefly in discussion of selection coefficients. And I said don't worry about them. This is the only time we're ever going to worry about them. So where do you think they're going to go in this equation? Where do they have to go, given what we just said? This is going to be a fraction. What's our numerator? It has to be, right? If we're saying mutation is increasing allele frequency, mu, the mutation rate, has to be in the numerator. As mu goes up, so does the allele frequency. Therefore, selection has to be on the bottom. This is specific to that homozygote. It ends up being the square root if it's a recessive mutation. The square root's not important. If it's dominant, then it's just mu over s. Where the square root came from is not important, but the difference is important, right? Allele frequency will be higher or change differently more rapidly in the dominant case because you don't have to wait around for two mutations to see the phenotype. You see the phenotype every individual mutation. So what is the allele frequency going to be? So this is the top hat question that we're not gonna end up doing. We'll do it, but we're not gonna answer it in top hat. So there's a mutation rate of a gene, which causes a achondroplasia, that's a human trait of dominant dwarfism. So it generates the affected allele at that rate. So people have measured mutation rate at the gene. This allele produces, it should say individuals who produce 74% fewer progeny. So that's the fitness metric. So we can do this. We can say, what's the equilibrium frequency? It's going to be the mutation rate over the selection. So 1 minus fitness. So if this is fitness, selection is 1 minus fitness. So we can predict mutation rate, we can measure fitness, we can predict the equilibrium frequency. You know what's really interesting about this example? That is the frequency of achondroplasia in human population, right? So we take different pieces of evidence. Let's measure mutation rate, let's measure fitness, let's make a prediction. That is, it's like they're independent pieces of evidence that actually corroborate the frequency of achondroplasia in human population. Not precisely, but that's because it's dominant. If it was recessive, the frequencies would be a lot lower. Right? We take the square root of this. But because it's dominant, we just treat it like this. Questions on that? Do you have to remember this? No. The point of the last 3 slides is that all populations are experiencing all of these simultaneously. You can do really cool stuff with it, like actually predict disease traits in humans with high precision, just based on kind of an intuition. Mutation keeping it up, selection keeping it up. Okay, so how should you think about approaching these problems? Make sure you can calculate allele frequency. Make sure you can go back and forth between genotype frequencies and allele frequencies both in and out of Hardy-Weinberg, right? Q squared, taking the square root of this, is Q only in Hardy-Weinberg equilibrium. Do not do that for a chi-square test, right? Where you're given numbers of individuals. Do not do that. What is Hardy-Weinberg? What are the kind of assumptions? And then we've broken all the assumptions and seen evolution in action. So recognize when you're dealing with this, right? When you're dealing with an allele versus a genotype. And I would add here when you're dealing with the next generation, right? F prime or Q prime, anything prime is the next generation. So you're going to be given all of these formulas, the mutation formula, migration formula, the huge table that describes fitness. Make sure you know when to use them. Hopefully the easiest way in my mind is you don't have to memorize all the formulas, you just have to memorize a couple really key components, right? Which one goes with which? Those are the only 3 letters you have to memorize. Yeah. Mutation with mu, migration with M, and fitness. If the question is asking you about fitness, look for equations that have a W in it. If the question is asking you about migration, look for equations that have an M in it. So that's kind of combining the last 2 ones. Make sure you know how to use these formulas. Make sure you know the abbreviations. You only need to know really those. I can say for a couple questions, one last announcement, but I don't think it's going to hit the target audience.